welcome everyone to another episode of Biology with Arseni. I'm your host, Arseni Aziz. So let's get started. Today, we're going to talk about proteins. Why proteins are so important, why proteins are so much needed in our body, and not only our body, why almost every living organism around um, Earth need these proteins, why they are so important specifically for us, what they are made of, what are their components, what are their monomers, how they form, everything we're gonna know today in today's episode. So let's go back to the drawing board, as we say. So proteins, so important, so nice to be around them. And as you can see there are lots and lots of sources for protein, from the red meat, from the white meat or the poultry, from the fish, eggs, dairy products, some vegetables, especially legumes, nuts, all sorts of stuffs that may have protein in them. So what makes protein so important? So as we know that proteins they are polymers, okay, and they are made of monomers, and the name of these monomers is called the amino acid. And amino acid have a very specific structure, okay. So from the looks we should know that what are the components that makes the amino acid. Now if we look over here now, we will find the structure of the amino acid itself. You'll find it around a center carbon atom, okay, connected into three stable, let's say molecules, or three stable groups, okay. On this side, you will find the amine group, the NH2 group, okay. So I'm gonna write it here in blue. This is the NH2 group. Okay, now this is amine group. This is where the amino acid gets its name. Okay, specifically amine, amino. So this is where the name comes from. On the other side, we have the carboxylic group, or the carboxylic acid. Okay, COOH. This side. Okay, this is the carboxylic group connected with a hydrogen atom, and down here we'll find a side group. Okay, this is a variable group, and we have tons of these side groups. Okay, we have different numbers of them and different shape of them that give every amino acid this distinct shape and distinct properties, and how this amino acid can react and how this amino acid can perform in almost everything around us. So this is the normal structure of the amino acid, how they made of, and we have lots of them. We have around 20, more than 20 amino acids around us. Okay, we can find it in so various forms and names. Here I'm mentioning some small names of them. We have something like called the glycine, serine, alanine, tryptophan, leucine, methionine, valine, lysine, and proline, etc. Now, this is how the amino acid looks like. Okay, now if you looked at that over here, for instance, let's take the valine for it. Okay, you find over here the amine group, you find the carboxylic acid group, or the carboxyl group. This is the hydrogen, and here this one in red. Okay, we will find this is the art. This is the side group, or the side chain. Okay, so let's see something more complicated like the tyrosine over here. Now, although you'll find it the same things, you find the amine group, you will find the hydrogen, you'll find the carboxylic acid, and you will find its side group. That's all of it. All of this is the side group. All of this is the R. Okay, all of that connected to a center carbon atom that's in over here and over here. You will find the center carbon atom, okay, in the middle, here and here and here, whatever I circle pink. this is the center carbon atom over here. So that's what makes the center carbon atom over here. And you will find that every amino acid has the distinct and has the same composition of hydrogen, amine group, carboxyl group. These three things are distinct. Now, the variable one is the side chain, of course. Okay, that almost give us 20, maybe more than 20 amino acids that form almost every kind of protein that we know. Okay, and of course that what we don't know yet. So this is our example of the amino acids. So we can say that the atoms in the proteins or in the amino acids, they can be carbon, they can nitrogen, oxygen, hydrogen, sometimes of phosphorus and sulfur. So, and as I told you, we have more than 20 amino acids that we know. Why they are important? Because they form lots of organ, lots of molecules. You can find it in meat, you can find it in fish, you can find it in poultry, legumes, nuts, and all other sorts of stuff of food. Knowing also, we have another fact that a 1 gram of protein support up to 4 calories. Okay, that is the same amount of energy that the carbohydrate gives. Okay, they form enzyme, they form hormones, they form supporting structure like the bone, the cartilages, the muscle, they form also hair and nail. They are also found in the buffering system in the blood to keep the pH of the blood stable at around pH of 7.4. That is so important for our cells to be able to live normally without any problem. And if it increased, we are in a case of alkalosis. If it decreased, we are in another case of acidosis. And both of these cases, well, they aren't the optimum for our cells, and they have their cons, of course, and they have all of their problems. We're going to talk about that later. And they also form the hemoglobin, which is an oxygen carrier, and the hemoglobin is the red thing that give our cells and give our blood the red color that we know. Now, also it forms protein gates at the cell membrane, which keeps molecules enter or leaving the cell. Okay, and in order to see where are these gates now, you can see these gates over here now, like this one I'm surrounding with red, and this another one which I'm surrounded also with red. Now these are proteins. Now you'll find here another protein that is hiding down here. Okay, and these proteins, it act like gate. Okay, like let's say like something that we call the sodium potassium pump, which helps in entering the sodium and the potassium inside and outside of the cell. Well, a gate like this, that form like this, it's made entirely out of protein. So proteins are important. And for the record, we are having this image from Encyclopedia Britannica. Shout out for them and for their excellent resources, very reliable resources that we can depend on anytime. So, how these amino acids bond together? Now, these amino acids bond together through something that we call a peptide bond. Now, the formation of a peptide bond depends mainly on a removing water molecule. Okay, removing water molecule is the same idea that how the monosaccharide in the carbohydrate, they are connected together. Okay, this is a dehydration reaction, and I'm gonna link the talk about the carbohydrates and anything around it for talks about the how the monosaccharide link together and how the water molecule is removed. You'll find it in the IAP there. If you are interested, go take a look. And if you reach over here, I guess you may help us by subscribing, liking like our videos, and check with your friend. It is for free, and of course you will provide knowledge to your friend, and it's going to be great. Now, this is how the polypeptide bond they are formed over here. Now we have three amino acids joined together by a peptide bond, and to say the specific to see what is happening over here. Now we have a hydrogen from the amine group and a hydroxide group from the next amino acid, the carboxylic group. Okay, and these are joined together and they form the peptide bond that you are seeing over here. And the removal, of course, of the poor water molecule that is left outside and is left by itself over there. Now, the protein itself, they can be found in four structures. Now, the first one is the primary structure, and the primary structure is a very simple one. It's just a sequence of amino acids connected together by a peptide bond, and they are forming a chain or something that we call the bead on a string or something. Okay, it seemingly sound this shape, and this shape is so simple, that shape is so easy, that has no problem to it, and it's so cool and so nice. And the second one is the secondary structure, okay? And the secondary structure is a folding of the primary structure, and it can be folded into two partitions. One of them is the alpha helix, which is coiling of the hydrogen bond between the carboxyl and the amine group, and it looks like this. I know my drawing is not the good of it, but that is how it looks, so we can consider this an amine group. And so I'm going to draw it not in green, I'm going to draw it in red. So let's say if this is an NH2, this is a COOH, and so this is an NH2, this is a COOH, and etc, etc, etc. Now, the other one is the beta pleated sheets, okay, and it can be formed either by a hydrogen bond or a disulfide bond. It's like a sheet that is a layer over layer over layer of each other, and it is so nice to have it, and it's so nicely shaped. Now, the third one is the tertiary structure, which is the most important structure because it is directly connected to the function of the protein and the enzyme, and most of the enzymes are found in this structure. And that tertiary structures look like this. It's so complicated. It's mostly let's say the complexity of the secondary structure, and it starts to bend over and it's shaped like this. And as I told you, it's most of the enzyme in this structure because it is directly related to the function of the protein. Now, the super arrangement of them is the quaternary structure, or you can call it a level 4 of protein folding. It's a very special arrangement. It's made of more than one type of the polypeptide chain, like, for instance, the hemoglobin. As you can see here, now this is the hemoglobin, okay? As you can see, this is an alpha 1, alpha 2, beta 1, and beta 2, and these are connected together and they are folded together. Now you have a different 4 types of peptide chains connected together to form the quaternary structure of the protein. Now, the protein can be classified, so let's zoom out over here. They can be classified into a simple protein, conjugated protein, and derived protein. Where did D go? I don't know where the D did go. So this is the derived protein. So let us go back to the simple protein. Now, the simple protein, when they are hydrolyzed, you can only find amino acid. You don't find anything associated other than the amino acid. So that's why we call it simple. It is plainly simple amino acid, and that's it. Like the protein albumin, globulins, the glutamines, the histones, the skeletal protein, or we can call it the fibrous protein, the keratin that we find it normally in the hair or in our fingernails, the elastin, and of course the collagen and the gelatin, collagen mainly. I guess people know that most of the celebrities use it to look youth and look younger, and they don't want to age or something, so I found it lame. The other one is the conjugated protein. Now, the conjugated protein is so meticulous. It is made from a protein part and a non-protein part. And that protein or that non-protein part is what determines um, the classification of that conjugated protein. So we have something called the cycloprotein, which is mainly protein plus carbohydrates, or the lipoproteins, which is mainly protein plus lipids. And if you are asking yourself, is there is any kind of interaction between the protein and lipid? Yes, in the lipoprotein, we have the low density lipoprotein, or the LDL, and the high density lipoprotein, or the HDL. And we can call both of them the good cholesterol, which is the LDL, and the bad cholesterol, which is the HDL. Also, we have the phosphoprotein, which is mainly protein plus phosphoric acid. And that's not only the classification of conjugated protein. We have metalloprotein also, which is mainly protein plus metal. One of the famous examples of the metalloprotein is the hemoglobin, which is mainly protein, which is the heme plus iron. Okay, so iron is the non-metallic, or I'm sorry, the non-protein part, which why makes us, when we have any kind of anemia or something, you'll find the doctor usually tells you you need to take some iron as a supplement. Okay, so that's why we need an iron, because it is needed in the formation of the hemoglobin. The other side is the drive protein, and when they are hydrolyzed or denatured, they give off simple or conjugated protein. Okay, and we have the proteases and peptones. That's the short story for them. There are lots and lots of the drive proteins, and we won't have any time for them. Maybe in another episode, and we will have to talk about these drive protein, because there are lots of things, and also we're gonna talk about the reactions of the protein and the enzyme, of course, how they function and what they do. And for the last thing of today's episode, how we test for protein. As we all know, testing is so important. If you want to test your food and know what kind of material are incorporated in that food you're feeding on, well, you need to test it. It's so important for the health organization to understand what the food is made of and why they are so important. And that gives us to the protein testing, which is the Pirate test. Now, the Pirate test is simply give an idea: is a peptide bonded presence, or there's a peptide chain presence, or no. That's it. Okay, so it is so simple, so direct. No heating is required. No water bath is required whatsoever. Pirate reagent is simply blue color, and in the presence of the protein, it turns into purple. And you can see it over here. This is the change. So purple on the other, on your right hand side is the Pirate test, and the blue one is the negative test. So that is all for today. Thank you so much for watching. Please, if you reach the end of this video, don't forget to subscribe, don't forget to like, to share your video with your friend. It is for free, and of course you will have someone who is in need for this information. I was Arseni Aziz, and that was Biology with Arseni. See you next time.